good afternoon and good evening, especially to our global immuno speaker today that is joining us live from Singapore. I'm Carla Roslin and I'm joining you today from my beloved Argentina. So it is a real pleasure to introduce our global immuno speaker, but before doing that, I would like to remind you that we will have Dr. Gabrielle Bells next week. And this is one of those global IMMUNOTALKs we're really going global live. So please stay tuned and join us next week for Gabrielle Bell's talk. Now, let me stop sharing this and get to introduce our global immuno speaker and co-organizer, Dr. Florent Ginhoux. It's a real pleasure to do this today. So, Florent was born in Bordeaux, France, and did his studies in biochemistry at the University of Pierre and Marie Curie. And he then went to obtain his master's degree in immunology from the Pasteur Institute and stayed in Paris, where he did his PhD studies, also at the University Pierre and Marie Curie. And I think it is during this time that Florent took his first steps in immunology. And specifically, he tried to understand the immune response to gene therapy, particularly in the context of gene therapy for muscular dystrophies. After his PhD, Florent moved from France to New York City in 2004, where he joined the laboratory of Dr. Miriam Merat, who was also a global immuno speaker. And this was at Mount Sinai. So this is where he did his postdoc, and he truly did a series of landmark discoveries during this time. It was because of Florent's work that we truly have a much better understanding of the origin and the development of microglia and dendritic cells. These studies really completely change our understanding on the ontogeny of these cells. And we now understand that, for instance, adult microglia have embryonic origin. As you can imagine, these seminal discoveries also launched his stellar independent scientific career. Florent stayed for a little bit in New York and became an assistant professor and member of the Immunology Institute at Mount Sinai in 2008. But soon after that, in 2009, he joined the Singapore Immunology Network, A-STAR, as a principal investigator and has been there ever since. He became an EMBO Young Investigator in 2013 and also became part of the GSK Elion and Black Immunology Catalyst sabbatical program. Now, Florent is an example par excellence of a global immunologist. He is not only a principal investigator in Singapore, he's also an adjunct visiting associate professor in the Shanghai Immunology Institute at Jiatong University since 2015, as well as an adjunct associate professor in the Translational Immunology Institute and Duke National University of Singapore since 2018. These laboratories around the world that he leads continue to focus on the ontogeny and differentiation of macrophages and dendritic cells. As you can imagine, Florent has received numerous recognitions for his scientific contributions. I want to highlight the fact that Florent has been a Web of Science highly cited researcher since 2016. This is a major, major accomplishment. I think we are all in for a treat. Florent, thanks so much for accepting our invitation to present, but also for accepting to be a co-organizer of these global IMMUNOTALKs. We very much look forward to your talk entitled Mapping the Mononuclear Phagocyte System Using Single Cell Technologies. Thanks so much.
Thank you very much. I think it's the longest and the nicest introduction I ever had. So it's really thank you very much for this kind introduction. Really. So thanks again for having me. And should I share my slides now? So should I?
Yes, but before we share, as you know, we love to ask our speakers, and you're not, not going to do this even if you're an organizer. So we'll have it.
I cannot escape it. Sorry. Okay.
Yes, no, not a problem, not a problem. Actually, on a more personal note, because we have shared interest, shared scientific interest, I had a chance to get to know you a little bit and realize how dynamic and committed you are to science. So I think we have a very appropriate question for you tonight, right? It's midnight in Singapore. So the question that we have is, what are the scientific questions that keep you up at night?
Okay, so yes, appropriately, clearly giving a global IMMUNOTALK keep me up at night, as you can see, although it's not too late. And just before I respond, I really want to congratulate you and Elina for this initiative that really allow immunology to reach out to all corners of the world. Although virtual seminar is not clearly my preferred format, they have their virtues, and clearly global IMMUNOTALK is one of them. So really thank you. So now back to science, just a quick answer, what keep me at night. I hope my seminar will show it. Myeloid cells keep bugging me. Of course, as you can see from my work, with a preference for dendritic cells and macrophage subpopulation. I think what intrigued me is their diversity, their plasticity, their complexity, and especially when you look at them in peripheral tissue, because a lot of immunology have been really focusing on lymphoid organs, spleen, lymph node. And I think like the last 10, 15 years, there have been this emergence of the realization that tissue immunity matters. And in fact, it's something that very few people did before. And it was clearly initiated by in fact, my mentor, Myriam Merah, that was the first really to start to look deeply in DC and macrophage, like Langerhans cells in the skin a long time ago. I think the way they develop their myriad of function that have been completely underestimated and also largely biased and simplified by a lot of in vitro assays. I think these are, compared to plenty of other cells that like T cells that people are focused on. So I guess there is still really a completely new universe and even galaxy to discover here. And I think once you get hooked to these cells, it's very difficult to stop. Although I allow myself sometimes to study some few like other cells, I think most of my work is focusing on this diversity of myeloid cells.
Fantastic. Thank you, Florent. Thanks for staying up at night and not only giving this talk, but keep on thinking about these fundamental questions. And we look forward to learning more about the diversity and tissue-specific functions of these cells. So you can share your screen now.
Yes, I will share.
And we can get started. Let's see. Can you? Perfect. Let's see.
Does it work?
Yes. Fantastic.
Okay. So, of course, again, thank you very much for the introduction and having me today. I think so. Today, I have the title is Mapping the Mononuclear Phagocyte System using sickle cell technology. I think I will give a different kind of talk that I classically give, and I wanted to go maybe a bit back in time, not too back in much, but like maybe 2015 since now. And I wanted to look back at how, in fact, looking at myeloid cells, mostly disease and after macrophage, and how using these like revolutionary tools like single cell technology, especially single cell RNA-seq, really have allow us to have a clear, different view and understanding of this system. So most of the work I'm going to present is coming from my lab in Singapore, in the Singapore Imaging Networks, my lab opened in 2009, but also from the lab in Shanghai, Institute of Immunology, that I opened in 2014-15, and also from the Transnational Immunoresearch, a single research, like also in Duke US, Singapore. So the focus of my work is, two related lineage, dendritic cells and macrophage. I'm pretty sure that we all know that all the attendants know that dendritic cells are the sentinel of the immune system. They are like called professional antigen-presenting cells, initiating and modulating T-cell response. Macrophage, as their name tells, are big eaters. Macro is big, phage is eaters. They are the scavenger of the immune system, but also a role that has been underappreciated and reappreciated in the last few years is their role in tissue function and tissue homeostasis. Again, not only during immune response, but during steady state. And I like to call them also like sentinel of tissue homeostasis to oppose them to DC, which are the sentinel of the immune system. And as I will show you later, like this, exquisite specific functional adaptation that macrophage, tissue-resistant macrophage have to each tissue where they reside. And of course, the aim is to really also understand through studying DC or macrophage, like a better understanding of tissue immunity as well as tissue homeostasis. And again, the big question is like, we want to understand where do they come from, ontogeny development, and how this dictate the function of the cells that in fact are the major player of what we call the mononuclear phagocyte system, the MPS that of course include dendritic cell, macrophage, and often another one, the monocyte that I didn't list in this slide. A bigger overarching question in the lab is, in fact, how these cells shape their cell identity during development and also in tissue. And this is a bigger question of like the integration of nature, where do you come from, versus nurture, where do you reside. Nurture can be a lot of factors, can be the tissue, but also can be inflammation and also like systemic signals that can be sensed by these cells. So the basic question that we are trying to ask, and in fact we are still trying to answer, and I think it will be probably endless, is where do these cells come from, ontogeny and differentiation pathway, and in fact how maybe do you come from, maybe detect some of your function, not all of them, but maybe some of them, and this is still a very important ongoing question in the lab. Of course, the better you know about cells, the better maybe you can manipulate, you can target, delete, amplify them, and we are aiming to manipulate, of course, this pathway of differentiation. And lastly, we are also interested in how conserve this pathway of ontogeny and function across species. And in the lab, we mostly work with mouse, human cohort, but also macaques, and also quite relevant to a recent pandemic. Although I will not show any data that on this is like bats, macrophage, and DC, which are quite unique compared to other species. So, you know, this slide shows you the classification of NPS, of the mononuclear phagocyte system, not based on phenotype, not based on function, but in fact based on where do they come from, what we call ontogeny across species. And this classification have really helped us to better understand the complexity of this system and to try to simplify it. And this allows us and in fact, this work was led by Martin Williams, which is a friend and a colleague, to try to categorize three main lineage in the MPS, of course, the dendritic cells, which are FLT3 positive cells, and I will go further in the sub-flavor of DC, the DC1, the DC2, the PDC. Of course, the macrophage, and I will also go into the fact that some macrophage here, the tissue and macrophage, have an embryonic origin, and this was kind of like a new factor that we took into consideration. And after this very mysterious like third lineage of monocyte-derived cells, that surprisingly, and I'm sorry for the ugly slide here, it just was a last minute change, that can give rise either mostly in steady state to what we call monocyte-derived macrophage, but also in inflammation, in disease, in stress to monocyte-derived DC. And this is really still puzzling us a lot. What is how this choice is taken, and what is the difference between monocyte-derived macrophage and embryonic macrophage, as well as monocyte-derived DC versus like FLT3 dependent DC. So this is like the overarching classification in terms of development of the cells that we are studying. And I will go back to this along the talk. For those who are interested, again, following this classification, also like Martin and I and plenty of other people have tried to also provide a common language to immunologists to try to identify and classify DC across tissue and across species. Because before this work, I think a lot of people were identifying DC mostly by phenotype, and this phenotype could change in species, of course, but also in tissues. And there was like a poor understanding of the commonalities across the different subsets identified in steady state and inflammation by different labs. So this kind of like this immunity resource provide really a way to identify disease in different tissue, in different species, to try to better like understand them. And just before going to data, I just wanted also to pass a few slides on single cell, because this will be like an angle also of the talk. And in fact, this was like kind of my feeling, and it's still a bit my feeling, although we grow a bit of maturity with single cell approach. But when we started single cell RNA-seq approach, this was exactly what I felt. I felt it looked like a miracle, because it's like an experiment where you're going to put some cells, single cell, and you will get a lot of data, a lot of clusters, and a lot of information. And this is really like this approach have really revolutionized really like the level of understanding that we have for like complex population. Of course, single-cell transcriptomic, have been really like evolving very, very rapidly across time. I think we started to hear, like, for example, this very nice review, like from Svensson, that showed a different approach, and I think we started to get into this game in 2013-14, and we used mostly this Fluidigm C1 chip that was like a low throughput chip, and I think now you can see that most of the lab in the world using like this 10x genomics that allow you to get a higher number of cells. And this is really scaling up. And this is only RNA-seq. And now there is more protocol to integrate also like epigenetics with ATAC-seq as well as site-seq to allow like the measurement of a lot of different antibody labeled with like oligonucleotide. I just wanted to remind also people that before you engage into single-cell RNA-seq, of course, be careful to have a question, formulate the question. Sometimes the miracle doesn't happen if there is no hypothesis. Of course, if you are like having like a more like atlas way of approach, of course, like just like doing a single cell of a unique tissue also is very democratic value in the fact that it can be shared with other lab and with the world, that we be able to also like download the data. I think what is surprising is when it's a targeted population approach, you need to have like somehow a prior knowledge of what you want to get and anticipate the data, because if you are focusing on a population and you expect heterogeneity, you have to have a certain level of understanding of how much heterogeneity you expect, because if it's 50-50, if you expect a 50-50 heterogeneity, it's quite easy with like 300 cells, but if it's a 1% progenitor cells, you better sequence more than 1,000 cells to get enough representation of your cells of interest. And it's always a balance between cell number and depth information, and of course, how big is your budget, because this approach are not so cheap, are quite expensive, and of course, a certain level of expertise to be acquired, and this was one of the bottlenecks for us, is the bioinformatic analysis that represent still quite a challenge. And just before I move back to the data, this was like a review that we, like Peter C., a postdoc in the lab, wrote with other like members of the lab to try to report on our experience through single cell and just like a small, like naive guide for immunologists, comparing the advantage and disadvantage of different type of approach. Looking at the C1 system, but also SmartSeq2 that have like higher resolution, but is quite costly, versus the 10x genomics. So all of these parameters are listed here, and the choice of them is based on all of these different parameters to take into consideration. So now back to the talk, and I will divide it in two parts. And a first part on dendritic cells heterogeneity, and a second part on macrophage. And of course, I hope that you all know that dendritic cells are, of course, antigen-presenting cells and very important cells that initiate immune response. Of course, they are here in between immunity and tolerance. I just want to also like highlight this picture that was taken by the late Ralph Steinman that discovered dendritic cells. This is the first picture of a disease from the mouse spleen in 1973. And this cell type really control immunity against pathogen and also make sure to maintain tolerance against self-antigen. And of course, learning about all of these two arms might allow you to design better vaccinal strategy, but also to understand how to treat maybe autoimmune disease that are basically a rupture of tolerance against self-antigen. I just wanted to remind also like how does it work? Diseases are often, and in fact, are found in every peripheral tissue in immature forms. They are constantly surveying their environments. Upon a challenge, inflammation, danger, destruction, they will basically sense these dangers. They will capture the antigen. They will migrate to the lymphatics. They will also engage in a parallel maturation program of activation that focus them to present antigen, and they will reach the lymphoid organ to speak with naive T cells, naive B cells, to initiate ensuing immune response, the adaptive arm. This is why we call a DC often the innate control of adaptive immunity, because within like minutes of hours after like inflammatory response, the role of DC is to understand what's going on, is to not only to catch what we call classically in the textbook of immunology, the antigen, the signal 1, but also to understand what's going on through sensing of innate immune receptors. This is called the signal 2, that will be able to translate and interpret what are the microbial products, the dangerous signal, to try to understand what type of immune response the system is facing. This will lead to presentation. Presentation on only signal 1 only lead to tolerance, ignorance, while signal 1 and 2, of course, this is oversimplified and lead to immune response. And often overlooked, but more and more like taken into consideration, is what we call the signal 3, which will be the type of cytokine, which is called like polarization, that will instruct the T cell what type of response they have to engage, and the classically known as Th1, Th2, Th17. And this cytokine polarization will also be instructed by the interpretation of the type of danger faced by innate immune receptors, such as toll-like receptors. So this is a general feature about DCs. Of course, the system is more complicated than this, and DC come in flavors. And I think, you know, for students listening to this, you are quite lucky, because we, in fact, Martin really led this project, and we really simplified, we really tried to simplify the field by, in fact, grouping all of different populations defined in different tissue or by different markers in different species into sub-lineage of DC. And we classify them again, not based on markers, but based on what do they need to become what they are. And we realized, for example, that all of this population of DC here listed, the CD8 in the mouse spleen, but also the lung CD103 in the mouse, but the CD141, CLEC9A, XCR1 DC in the blood in human, in fact, are all cells that are BATF3 or even now IRF8 dependent, and we classify them as DC1. The CDC2 are IRF4 dependent and include the CD4s in the spleen, but also the C7b in the tissue, as well as C7c positive DC in human. And of course, the PDC lineage that I will also like allude to, because there is a bit of concern about the DC nature of, in fact, the plasma cell to dendritic cell lineage. But regardless, PDC are E2 dependent and arise from like a dedicated progenitors. And I will go back to this in more details. So this was like a ontogenic classification of DC subsets. What is great is it's also linked, ontogeny seems to be also linked to like conserve function, and DC1, for example, seems to be the DC lineage that are specialized in cross-presentation, but also inducing Th1 response, while DC2 seems to like be more specialized in Th2 and Th17 induction. And of course, PDC are very well known to be interferon alpha secreting cells, and of course, they're a very important role in antiviral response. So in terms of evolution here, you can see that the DC1s, through their cross-presenting Th1 function, might be specialized and might represent a DC lineage which is specialized to deal with intracellular pathogen, because Th1 and cross-presentation is exactly what you need to induce to fight virus or intracellular bacteria, while DC2 seems to be more specialized into extracellular pathogens through the induction of Th2 and Th17 response. Of course, this is an oversimplification, but it gives you a framework of thinking, in fact, why do we need DC subset? And I think this may be the explanation of like having a specialization of a type of response. And this specialization, what is interesting, is even intrinsically endowed in the cells, because what is very intriguing is DC1, even before encountering a pathogen, already expressed, for example, mRNA coding for IL-12, while the DC2 already expressed sometime in tissue mRNA coding for IL-23. And this population, this mRNA, are the signal needed, for example, IL-12 for Th1 or IL-23 for Th17. So it seems that the DC lineage already knows to which type of T cell like induction they are specialized for. And I think in terms of evolution, it makes so much sense, because the DC1 and DC2 express different type of like sensing receptors that have maybe specialized into wiring toward a type of response. And basically for us, we believe that this lineage represents somehow what we call anticipatory immunity in terms of class of pathogen. So now, why spending so much time about this? Because if you believe it, you can make the parallel with T cell. It's like, you know, lot of immunologists have studied a lot like how a T cell progenitor becomes specialized to become CD4 or CD8. And I just wanted to make the parallel here between DC1 and DC2. And this question, in fact, never really interested anybody since like the last, for a long time, at which stage of differentiation across this differentiation path from the common DC progenitor to the circulating DC precursor, the pre-DC, when is this commitment to DC1 and DC2 is taken? And it's quite important to know it, knowing that these cells are specialized to a certain type of response. And in fact, this question was led by Andreas Schwitzer, which was a postdoc in the lab at the time. And in fact, at the time in the field, in fact, we didn't know so much. And the idea was that neither of these precursors in the bone marrow, the common DC progenitor or the pre-DC, which is a circulating DC precursor, are committed to any of this lineage, and both of them can give rise to both DC1, DC2, and PDC. So this will be the simple hypothesis when you don't know anything, that they're all equal and one progenitor for all DC subsets. But Andreas had a different idea. He had this hypothesis that maybe already early CDP or even pre-DC could express and could have this kind of transcriptomic imprinting to give rise either to DC2 or DC1. They could be like prime toward of this lineage. And exactly this, I want to highlight this because this is exactly when you need single cell approach, because if you don't know any markers of this population, how can you make the discrimination between this DC2 prime versus DC1 prime DC progenitors? You need really to sequence them unbiasedly at the single cell level and really look in their transcriptome. And this is exactly where we really wanted to map the heterogeneity of DC precursor progenitors. So looking at the MDP, which is a monocyte DC progenitor, the CDP, the common DC progenitor, which is committed only to DC generation, and the pre-classical DC that give rise to DC1 and DC2. And at the time, we use the C1 fluidine solution that was like plate of 96 well. And what we did, we sorted the MDPs, the CDP, and the pre-DC, and we load them in the plates, and we isolate each cells from each population at the single cell level, and look and profile the mRNA transcriptome. Sounds simple, but at the time it was for us a big challenge, and it was kind of like a very, very difficult project, and especially the initial bioinformatic work that have been quite challenging, but we managed. And this simple graph really really highlight the major finding of this is that we could see emergence of DC subset commitments very early, in fact, in the bone marrow. So of course, just looking at this work, so what we do here, this is the MDP, the CDP, the pre-DC, MDP, CDP, pre-DC, which are aligned along the differentiation potential. And what we do is we project on each cells that have been sequenced a DC versus a macrophage signatures. So the MDP is a monocyte macrophage DC precursor. So you can see some cells that already have like a DC versus a macrophage signatures. And of course, the CDP, which is a DC precursor, mostly have DC signatures, and the pre-DC, which is a circulating DC precursor, all of them are committed to DC. So this was some kind of a positive control to show that our approach of projecting signature worked. And this is, again, the major finding of the paper. Looking at pre-DC here, we could see, and even at some few CDP, we could see some DC2 versus CD1 pre-committed pre-DC, showing that already in the bone marrow, before exiting to the blood and the tissue, some of these precursors exhibited some DC lineage, sublineage commitments. Of course, you as immunologists, when you see these cells, we did, okay, let's try to find a marker of these cells versus this one. So we extracted the data from this as DC2 committed, DC1 committed, and uncommitted, and we plotted the gene expression. These are the uncommitted, these are the DC1 committed, DC2 committed. And we could see that, in the DC2 committed, the emerged expression of certain markers, extracellular marker like Ly6T, Ly6C, or CD24 in the DC1, for example, here. So this marker was selected and tested. And when we look then at the pre-DC population, we could then split this pre-DC population in 4 populations based on C-glycage and Ly6C. And I don't want to go too much in details, but what happened is that, in fact, the pre-DC that we sought was a homogeneous population, and this was published in 2015, was composed of subpopulation, which like a predetermined commitment, and the identification of a Ly6C positive pre-DC stage, which was DC2 committed, as well as like a CD24 XCR1, we call it double negative, but it's also expressed CD24, pre-DC, and the Ken Murphy lab also at the same time published with us and identified c-KIT as a marker of this DC1 committed pre-DC. So single cell really allow us to identify more complexity in this like a continuum of differentiation to dendritic cells. And again, what's interesting is like we identify this single H pre-DC that still have this pre-DC potential. And this is a transition to also like introduce some work that we have done asking, where PDC come from. PDC ontogeny and PDC nature are always been a matter of debate. And the question is like, are really PDC really belonging to the DC family? Because they don't really behave too much like as DCs. They don't really present very well. They need to be activated, and they don't like have this kind of polarization capacity, like in terms of initiating and polarizing T-cell response. And I want to highlight the work of like the group of Roxane Tuziwan that have clearly showed that the major PDC in fact do not come from a classical myeloid precursor like the CDP, but come from a common lymphoid precursor, the CLP. The work also from Dominique Grund through like a massive like single cell approach and computing approach have identified also maybe some shared ontogeny between B cells and PDCs. And our own work, where we from Regine Dress, that really like have like systematically using the C1 single cell approach, sorting each population that you can see. So we have sorted like the LMPP, which are this early like progenitor, the GMP, which are the granulocyte macrophage monocyte progenitor, the CLP, which is more lymphoid, and all of these different stages of DC differentiation, and again projected either what we call a PDC versus a DC signatures. And in a nutshell, the major finding of this paper is that the CLP already exhibit a significant commitment, at least at the transcriptomic level, toward the PDC. So from this work and plenty of other complementary experiments, we concluded that maybe PDC could arise mostly from CLP through like an independent lineage compared to the classical DC that arise from like the CDP. And again, this raised the question, of course, what are really PDC if they don't share common precursors with DC, and especially sharing a common origin maybe with B cells, and they could represent some kind of innate lymphoid cells, like that, of course, a name could be discussed like in the future. But for us, it's quite important to realize that in ontogeny, most of PDC doesn't seem to arise from the same ontogenical pathway than classical dendritic cells. So this is an updated differentiation pathway, a map, where you can see now that most PDC in the major population derive from this Ly6D positive precursor arising from CLP. The DC arise from the CDP. Although I want to highlight that I think what is still like a matter of debate in the field, and this is the major contribution of the paper from Roxane Tuziwan, is that she was still able to show that a minor population of PDC could arise from this CDP. But now the real question is like, are these cells, PDC because they express the same marker in PDC, or could they represent in fact a different lineage? And I think this is like an ongoing question in the lab, and I think will be very exciting to follow up on this question. I also wanted to, before moving to human data, is to highlight a paper from the lab from Dr. Baptiste Janela from 2019, where he has shown that sometimes DC are only considered to be antigen-presenting cells. And in this paper, Baptiste have clearly showed that in a model of skin infection, DC1, even before migrating to lymph node, play a very important role in mobilizing and activating neutrophils through like VEGF secretion, and the absence of this DC1 and the absence of VEGF alpha secreted by DC1 led to like an aborted neutrophilic activation and response. And this is also was perfectly in line with the report from Carlos Desfresno and David Sancho group showing that GNGR1, which is like a DC1 marker, really limit tissue damage by dampening neutrophil recruitment. And both of these papers have been also, I like the important crosstalk, in fact, between subset of DC, like the DC1 and neutrophil. And it's quite important also to follow up in the future. So what about human DC progenitors and our experience also on trying to map this DC progenitor, but now in human, in the blood, was a very important collaboration with Dr. Joachim Schulze from the Limes in Bonn, Germany, where he helped us at the time to map a DC subset in the blood. And you know, the difference compared to the mouse. In the mouse, we knew the progenitor in the bone marrow. In human blood, we didn't know where to look except looking into FLT3 positive cells, which FLT3 mapping basically had to be expressed by DC and their progenitors. And I don't want too much to go into details, but this approach, along through the sequencing of like more than 1,000 cells, allow us to identify a population of cells that we call pre-DC that were phenotypically similar to PDC. Exactly the same marker of PDC, like a 3D303, 3D304, which are really classical marker of PDC, were also expressed by pre-DC, the DC precursors, 1, 2, 3, and slightly higher level of CRNC. So this observation really highlighted that maybe a lot of previous study defining PDC with this specific, this marker may have also included pre-DC, and this why it could explain why people have also reported the fact that PDC could represent precursors of DC, of classical DC, just because the PDC preparation could be contaminated by, in fact, simply DC precursors. So when we look at pre-DC and look at their canonical function, we could see that, you know, of course, they were not able to secrete interferon alpha, but were very, at least able to secrete polarization cytokine like IL-12, while PDC could not. In terms of like T-cell proliferation in an allo-MLR type of approach, pre-DC were like quite potent at stimulating T-cell, while PDC were not. And also in terms of morphology, like pre-DC have this unique, in fact, like typical DC morphology, while PDC have like, again, this like a typical round shape with like this polarized, like a reticular endoplasmic. So really highlighting the clear difference between this pre-DC and this PDC. So this paper was led by Peter C. and Charles-Antoine Duterre and Jimmy Chen in 2017, mapping the human DC lineage, like from the CDP, the pre-DC, and again, like in the mouse, the identification of like DC committed precursor, like the pre-DC2 and the pre-DC1, that can now be identified by using different markers. And now the aim is to look at how this commitment and how this proportion of these cells change, maybe during disease and inflammation. A side story, just to highlight that, you know, identifying pre-DC or subpopulation is just not a stem collection exercise, or just not to only publish. It have also like some biological relevance. What we noted very quickly is that the pre-DC in human blood, not in tissue, but in human blood, pre-DC had the highest level of expression of CD1, which is CD169. And we know that CD161 is an HIV receptor. That led me to contact immediately my friend and colleague Philippe Benaroche from Institut Curie and ask him, can you test maybe if pre-DC could be efficiently infectable with HIV? And this is what we have done. And we compare the capacity of these different populations, DC1, DC2, PDC. And you can see here that primary DC in human blood are poorly infectable by HIV. DC1 and PDC are not infectable. CDC2 at low amounts, but of course, and surprisingly and excitingly, pre-DC, at least in proportion, were more able to be infected by HIV. And this infection and capacity of the cells to produce functional virus, measured at P24 positive pre-DC, could be also abrogated by incubation with an anti-Siglec-1 antibody, showing that Siglec-1 on pre-DC is important for the capture and after the internalization of the virus. So this really exemplifies how, like, the more you know about, you know, population, and the more you can make, like, you know, in fact, random discovery that may have like some biological relevance. So I'm going to stop here for the DC part and move to our work on macrophage heterogeneity. And I want to discuss about what we call the cardinal points of macrophage biology, and really what are the factors that control what we call the resident tissue macrophage identity. And this is a very nice, like effort from Dr. Camille Blériot that I've really put together this review in Immunity last year. And we listed as 4 major parameters, factors that could modulate and play an important role in shaping tissue macrophage identity, starting with origin, where do they come from, and we'll come back to this, and where do they reside, local environments, maybe, and something that I will have not too much time to discuss is, in fact, how much time they are spending in tissue, and of course, inflammation, how a change in environment, infection, or other like inflammation may change their identity. So let me start first with the local, and of course, really, this is to insist that this complexity of cues that macrophage sense constantly in tissue cannot be explained by a simple like M1, M2 type of polarization that is often too much used in biology. So let me start with local environments. And just to highlight again that tissue and macrophage are a fundamental element of all tissue. Of course, as immunologists, we mostly focus on their role on immunity and resolution of inflammation, by the fact that they can recognize pathogen and react and play a role in inflammatory response. But as you can see here, in every tissue, in steady state, in homeostasis, macrophage play a very unique role. For example, in the lung, alveolar macrophage are very important for surfactant clearance, and absence of alveolar macrophage or disruption of this clearance lead to a disease, alveolar proteinosis. In the bone, osteoclasts are important to remodel the bone, and failure of macrophage to do so lead to osteopetrosis. And those are functions which are in steady state during development and independent of any immune function. How does this macrophage acquire this unique function? Is in fact through the expression of crucial what we call transcription factors. Of course, womb expression is induced by the local environment, and there has been a lot of effort by the lab of Ido Hamid, Miriam Merad, Martin Williams, and also Chris Glass, and plenty of other people to really identify in each tissue and macrophage what are the combination of transcription factors that are unique to this macrophage. And for example, in cover cells, people have identified ID3, spicy, LCR alpha to be unique. In alveolar macrophage, is PPR gamma, BAC2, CBP beta. In macroglia, SAL1, SMAD2, SMAD3, etc., etc. And really, this tissue-resident macrophage program, which is specific to tissue, is again induced by the macroenvironments. And for us, this is kind of like the rationale also for this study that I want to share with you, that was led by Dr. Sedovets-Shakarov. Because, for example, in the lung, most of people look at alveolar macrophage, which are in the alveoli, in fact, outside the body, like in these structures. But there is also another population, a minor population of macrophage, which are in the parenchyma, the interstitial macrophage that you can see here. And in fact, nothing really is quite known about this macrophage population. And this is how we started to look at them. Trying to identify them. So this is how we do. We take the mouse lung, we exclude the monocytes, we exclude lineage, we gate on what we call MERTK64, which are defining all the macrophage population of the lung, and Siglec-F versus 7B will allow you to separate alveolar macrophage, which are Siglec-F positive, versus this minor population of interstitial macrophage, which are 7B positive. So we apply single-cell RNA-seq to this interstitial macrophage to understand how homogeneous or how heterogeneous this population were. And in fact, it was really one of like the most like striking and really like easy results. Is like very quickly we realized that this population were quite heterogeneous, and in fact were composed of two populations and clearly independent population. And you can see this blue cluster here that express CX3ACR1 or MHC class 2 genes, and this cluster in red that express like a more macrophagy gene like PLOR, TGF-beta, ICAM2, CLEC4. And this is like the projection also of the other marker of this cluster. Of course, this is single cell, so we wanted to have a better resolution. And clearly, when we gated on this population, we could see, as predicted by RNA-seq, single-cell RNA-seq, that class 2 was maybe a good proxy for separation. So we sorted intercellular macrophage by MHC class 2, based on MHC class 2, to perform bulk sequencing in order to identify better, to have a deeper resolution. And again, we could separate the population based again on 63CR1 versus class 2. And surprisingly, in this bulk experiment, we spotted this marker called LIVE1 here, which is the hyaluronic acid receptor. And when we validated this by flow, we could identify among the intercellular macrophage clearly two populations of live positive and MHC class positive intercellular macrophages. And we confirm also that 63CR1 were highly expressed on the class 2 high population. So through the use of single-cell RNA-seq, we could identify two independent populations of intercellular macrophages. So what do they, what are they, what do they correspond to? And in a nutshell, to recap the paper, we identified that both populations arise, of course, I will come back to this, arise initially from fetal progenitor, but a long time will be slowly replaced by monocytes. And when monocytes arrive in the lung interstitium, but also in other tissue, we look at fats, we look at the heart, we look at really multiple tissue that have these similarities. We could see that monocytes could give rise to these live positive macrophage that were associated to blood vessels, and especially to blood vessels, in fact, uniquely blood vessels that have smooth muscle cells. And in fact, there is this unique interaction between live positive macrophage endothelial cells and smooth muscle cells. And this was also highlighted in this paper from Dr. Véronique Angeli the year before. And the other flavor of this monocyte-derived intercellular macrophage was the MHC class 2 high, 61 high, but live negative macrophage that were more parenchymal, more inside the parenchyma, but also often interacting with nerve bundles or nerve endings. So again, this work highlighted not only the fact that intercellular macrophage can be heterogeneous, but this heterogeneity is likely also determined by the niche where they reside, like the blood vessels versus the nerves. And of course, functionally, this was quite interesting to see that upon depletion, and we had this mouse model that allowed us to deplete like live positive macrophage. In a model of like lung fibrosis, we were able to show that that macrophage, live positive macrophage, in fact, control the extent of fibrosis and limit the recruitment of inflammatory cells like neutrophils or monocyte-derived cells, and have this kind of like inhibitory role in terms of like tissue permeability and also like tissue inflammatory cells recruitment. So just to add like a bit more complexity into this scheme is that monocyte-derived cells in steady state, of course, can give rise to macrophage, but also can give rise in multiple tissue to two subflavors of macrophage: the live positive, which are associated to blood vessels, and the live negative, which are associated often to nerves and bundles or endings. So before I'm almost done, I just wanted to speak a little bit about origin, which is also a major focus of the lab, and also like for us like a very important point of macrophage biology. And this interest stemmed from the work I've done with Myriam Merad, where we discovered together that macroglia, the brain-resident macrophage, in fact, arise not from monocytes, but arise from these very unique progenitors during development. In fact, during very early development, around E9.5, 10.5, yolk sac macrophage, which are macrophage made in yolk sac, and we'll show you later what is the yolk sac, migrates through the blood vessels and enter the cephalic mesenchyme, and will migrate into the neuroepithelium very early and give rise to macroglia. And that will just like stay there, in fact, forever until adulthood. And this is to illustrate a lot of multiple years of research where we have shown that during a lot of macrophage population, in fact, all macrophage population initially arise from embryonic precursors. And there is two waves: the yolk sac macrophage, which are coming from the yolk sac blood island. This is the primitive hematopoiesis, which is semi-independent. It was shown by Frederick Geisman. And this yolk sac macrophage enter the blood circulation and will colonize the brain rudiment, but also every other fetal organ, including the skin, but also the lung, the kidney, and the liver. Of course, the system is a bit more complex. There is also another site later of hematopoiesis, which is called the definitive or semi-definitive, semi-dependent hematopoiesis in the fetal liver. I don't want to go into details where this progeny come from, but there is generation of like fetal liver monocyte that also migrate into tissue and give rise to fetal macrophage. And there is this like a different tissue-specific contribution of like yolk sac macrophage versus fetal liver, and later on bone marrow monocytes. So this is what I want to recapitulate here. Is that here, like trying to see that there is macrophage heterogeneity in origin and maybe in function. So in the brain, most macrophage come from the yolk sac. Langerhans cells initially, like lung, alveolar macrophage, liver, couple cells, initially arise from the yolk sac, but are replaced by fetal liver monocyte. But this will stay like this until adulthood, although there is some controversial data on alveolar macrophage that we really need to readdress. And in the contrast, some other organs also arise, the first seeding is from yolk sac, they are replaced by the fetal liver, but after birth, some of them will be replaced either slower or faster by bone marrow monocyte. And at the time here, you can see that we draw a complete replacement. And I just want to highlight that, in fact, we were not exactly correct. And I just want to show you later how did we realize that we were not correct. In fact, the fact that monocytes are not completely replacing fetal macrophages. And we realized this by using this model, and which is what we call the MS4S3 fate mapper of monocyte. And this is beautiful work initiated by Amanda and also finalized by Zhaoyan Liu, a postdoc in the lab in Shanghai, that was really trying to look for a gene expressed only by monocyte progenitors and not by monocyte themselves, not by macrophage, not by DC, in order to make what we call fate mappers to really pinpoint which macrophage come from monocyte. So the idea, if this is the scheme of differentiation of monocyte arise from GMP, MDP, CMOP, and they give rise to monocyte. So the idea was to really find a gene only expressed by CMOP. So if you build like a Cree fate mapper, the Cree will be only initiated at this stage and will be shut down, and you can after follow and track the progeny of this population. And then, allow to map monocyte contribution against DC, maybe, and against also yolk sac macrophage. And again, using single cell, it was able to map what are the cells, the progenitors, the bone marrow, which are committed to monocyte versus DC. I don't want to go into details, but we look at the gene expressed only by monocyte committed progenitor versus DC committed progenitors, and we came out with a list of genes, including, here you cannot see, but MS4S3, that were like quite specifically expressed only in the monocyte prime progenitors and not in the DC prime. This is another representation where you can see here like the CMOP, which are the monocyte progenitors, and you can see the quite specific expression of MS4S3, mostly in CMOP. So this really convinced us that MS4S3 was the right genes to go for and build like a Cree fate mapper. And this is what we have done. We have inserted the Cree IRS in the 3' UTR of the MS4S3 locus, bred it with the ROSA reporter. And after look at what did we fate map. So here, there were like a very good news, but also initially like a sad news, but the fact that we managed to deal with is, of course, we were very happy to see that monocyte were very specifically labeled compared to DC and other population. But we realized that, in fact, granulocyte here was also labeled. And in fact, we have done, as you can see here, not a CMOP fate mapper, but a GMP fate mapper. So MS4S3 is also expressed on GMP, and this allows the labeling of both monocytes and granulocyte. But in fact, it was not such a bad thing in the sense that granulocyte and monocyte lineage are quite easily distinguishable by multiple approach. And then looking at this model, and this slide, in fact, recapitulates 10 years of work from different labs where we're able to compare what is the monocyte contribution to tissues in macrophage just by measuring the tomato labeling in macrophage. So of course, microbia being from yolk sac macrophage and not from monocytes, there were no tomato labeling. Langerhans cells were similarly the same, sphenic macrophage the same, while the other population, the dermis, the gut, kidney, had like a significant a tomato labeling in accordance to previous work. So now I want to highlight something that where we were incorrect is that you realize here that at 20 weeks, for example, the dermis and the gut show like a plateau. And this means that there will be no further contribution of monocytes. So it means that some macrophage arising from the embryo, because here you can see they're tomatonegative, will maintain themselves until adulthood and onto the lifetime of the mouse. And this allows us to, in fact, to correct our model instead of like really putting a full replacement as before, is to putting a partial replacement in this tissue. So in the gut, in the dermis, most macrophage will derive from monocyte and monocyte will replace the past contribution from embryonic precursors, but there will still be a minor population of macrophage of the embryo that will stay and remains until lifetime. So now this led to a very intriguing question of, what is the function of this embryonic versus adult macrophage? And this is something that the lab is following. And I'm almost done. This is my last two slides. Speaking about time, just to let you know that, this is the way we are moving about time, is just using this model, trying to understand, in fact, what's going on in a tumor, where maybe, just to push the idea that a tumor might be composed of different types of macrophage, not only the embryonic one, but also some monocyte-derived one that have been recruited before or during the tumorigenesis. And this is what now we're trying to understand, is the heterogeneity of tumor-associated macrophage in terms of origin, but also in time of time of recruitment. So again, ontogeny time, but also maybe probably sublocation in the complex tumor environment might matter. And this hopefully will be the matter of my next global IMMUNOTALK. And with this, I want to thank the people which have done all of this work. So I highlighted the work of the postdoc in the lab, Svetoslav Shakarov, for the long work, Camille for the review, Charles-Antoine Duterte. I didn't have time to present his work, sadly, here. Zhaoyan Liu for the MS4S3 mouse, and also all lab members and core platforms members of the different institutions and close collaborators, including Véronique Angeli here. And I thank you very much for your attention.
Thank you so much, Florent. Thanks for sharing your very...
I hope I didn't go too much over time, sorry.
No, it's perfect. We are perfect on time. Thanks for sharing your very elegant work and truly for enhancing the precision by which now we can understand the complexity of all these cells. So thank you so much. Now, before we leave, let me share, let's see. I want to remind people how they can ask questions. So if you go to our Global IMMUNOTALK Twitter account, you will already be able to find a tweet that says ask questions for Dr. Florent Ginhoux here. Please remember, you can ask your questions by replying to this tweet with, of course, your question and mentioning hashtag Global Immuno and the account that Florent will use, which is his own personal account at fginhoux. So thanks, Florent, especially for connecting so late. And thanks everybody for joining. And we look forward to seeing you again next week with Gabrielle Bell's talk. Bye, everybody. Bye, Florent. Bye.